therapy. And cellular membrane replacement therapy, it's like we have hormone replacement therapy, cellular membrane replacement therapy. It's like when you have in your house, you have roof that you need to renew it because it's dripping. Well, the cell membranes, whether your cell membrane itself or the mitochondrial membranes, that's why I call it cellular membranes, all the membranes of the cells, whether it is the organelles, membranes, or even that needs to be renewed, and you need to provide them with the supplements that helps to renew quality membranes. Same thing when you are resurfacing your roofs of your house. But when you're going to do that, is that something you start with in the beginning when you're treating the patient? No, I would do the metabolic, fix the immune of the patients, fix the redox, and then before I do exosomes and regenerative, I will start doing the membrane replacement therapy. So hormone replacement therapy has to be first. And then after you do the three pillars, which is the immune, metabolic, and redox, you need to fix those, screen it. And then you go ahead before you do regenerative, because I can think it's part of metabolic and regenerative, the membrane replacement therapy. And then you start doing that, and then you do the exosomes for regenerations and healing. So you start with detoxing, and then repairing, and then regenerating. I think that's the three steps. But we have Dr. Garth here. Nicolson can confirm what we just said. Now it's CME sessions. We cannot talk about business anymore. But after we finish the CME, everybody can express his whatever he wants. So go ahead, Dr. Garth. The mic is yours.
Okay. Let me see if I can share my slides with you. Okay. I don't see, yeah, here we are. And I think I've got to turn this on here to presentation mode. I don't know what that means. So I'm trying to share. Okay, for some reason it's not sharing. So I don't know, because there's so many there to share or what, but
can anybody guide him through? Garth Nicolson, can somebody help him out?
Can you hit the sharing button on the bottom, Garth?
yeah, I did that.
Is it allowing you to share?
It says it's failed to start.
Dr. Haas, make him a co-host. You should be able to share it then.
He is already a co-host.
Maybe there's
he should be able to.
Maybe there's something in the settings that's not allowing him to share. Sometimes, I mean, that happened to me at the beginning.
Right.
Okay,
Otherwise,
so let me hit
share this lecture with Sheldon, and then Sheldon.
Let me hit the presentation button again. Okay. And now let's try to share it. No, it's not starting it for some reason.
What about share screen? Share screen and then
screens.
Just share the screen, or if you push share and then
there
you go. Yeah, you got it.
Okay. Okay. As I said before, I'm Garth Nicolson. I'm a professor emeritus of pathology and laboratory medicine while I was at the University of Texas in Houston. And now it's not advancing for some reason. Okay. So I'm going to talk about basically membrane structure and dynamics in the fluid mosaic membrane. And the reason this is important, and you've been touching on this during your conversation, is that our membranes have to remain fluid. If they don't remain fluid, essentially you die. So the fluidness is necessary, and as you age, it actually becomes less fluid. We were talking about the endothelium and how it's necessary to maintain its ability to stretch and contract and so on and so forth, and that requires a fluid membrane. So to do that and do a number of other things, we've come up with the membrane lipid replacement. And I'm going to talk a lot on mitochondrial function and the repair of mitochondrial membranes, because mitochondria supply the energy that does essentially everything. And as you age, basically your mitochondrial function goes down. So it's a very important aspect of aging. And we'll talk about that as well. And then I'll talk about membrane lipid replacement for reducing symptoms. For example, like fatigue, pain, and a variety of other symptoms. And we've been studying this in a variety of different chronic illnesses, and I'll touch upon that. And recently I've done some work on chemically exposed veterans. And the nice thing about membrane lipid replacement is it will slowly partition out hydrophobic molecules from your cells. And we were discussing this generally a little bit earlier. And this is very important because these toxic molecules build up in your cells and they're stored in your cells, and it's very difficult to remove them. And this is one method that can do it, but it's very slow. But that has an advantage, as I'll get into. Okay, I'm going to talk about membranes a little bit, and this goes back 50 years when I was in graduate school, actually. And at that time, the model of membranes was this tri-layer model put forward by Daniele and Dobson, where there's protein lipid sandwich, proteins on the outside, lipid bilayer in the middle, and there's a competing model called the lipoprotein unit model, which were just lipoproteins bound together to form a monolayer. And there were a number of reasons why this was wrong, and I'm not going to go into that, but essentially, we have to have a lipid bilayer in membranes because it's been shown the membranes have a lipid bilayer. Proteins are globular, and it can't have that with the Daniele-Dobson model. And they're transmembrane proteins. They can actually go through the membrane, and that's not really shown here. They're domain structures of membranes. I'll get back into that because it's very important. Membranes are asymmetric, and there's independent movement of proteins and lipids. You can't have that here, and you can't have that here. But over here with the fluid mosaic model, all of these criteria are met. So this is why the fluid mosaic model became the model for membrane structure about 50 years ago, and it's remained ever since. And this was the publication that was the most highly cited in all fields of science for several years in a row. And the reason that this was so important is because it fulfilled the thermodynamic properties of membranes. It had a lipid bilayer, but it had globular proteins that some of them went through the bilayer completely, others did not. They were both integral and peripheral proteins, meaning that they were proteins loosely attached to the membrane, as well as those that were integrated into the lipid bilayer of the membrane. And there was mobility of membrane components, which is extremely important. And as you age, that mobility is impaired. And there's maintenance of membrane asymmetry, which is present for this type of membrane. So these are now the textbook models of membrane. They're all based on the fluid mosaic membrane model. Essentially, every 100% of the models in all the textbooks are now the fluid mosaic membrane model. These are just some examples of that. And I've written a number of reviews since that first one, which was so highly cited. Here's a more recent one where I touched upon the importance of the fluid mosaic model and membrane lipid replacement, which is my main topic tonight, because this is what we're really going to talk about. Because membrane lipid replacement, and we use oral membrane lipid replacement, is the use of membrane glycerophospholipids. So these are the normal type of phospholipids that you hear about associated with membranes, like phosphatidylcholine, phosphatidylethanolamine, phosphatidylserine, phosphatidylinositol, and phosphatidylglycerol. Now, it's important that we have a mixture of these and not just, for example, phosphatidylcholine, like some of the treatments use, because some of these are important precursors. Like, for example, phosphatidylglycerol is an important precursor for cardiolipin, which is absolutely essential. So also, it's important to have fatty acids, which are essentially unsaturated, to help restore the fluidity of membranes. So we have a a product, and John Casey is on board here tonight, and his company makes this in a commercial form called NT factor lipids. But essentially, it's a mixture of glycerophospholipids. But it's important difference here between that and just lipids is these are protected with fructooligosaccharides, a very small dose of inulins, plant inulins, protect the lipids, protect them from enzymatic damage while they're taken through the gut, because they're really taken into our system in the small intestine. It also protects against environmental damage to these, oxidative damage, for example, or heat, or freezing, or whatever. This is what plants use to protect their lipids, to protect their membranes. That's why plants like trees can survive even when they're completely coated with ice and snow and stuff. They can still survive if they have inulins to protect their membranes. So membrane lipid replacement has been used in various formulations with a number of other components. Some I'll talk about additional supplements for mitochondrial function, and I'll touch upon that. But at the end of my talk, I'm going to go pretty heavily into the use of the membrane lipid replacement for removing toxic hydrophobic molecules from your system. So these could be, for example, hydrophobic toxins from mold. They could be hydrophobic chemicals like petrochemicals that I'll show you a study with Gulf War veterans who were compromised because of their petrochemical exposures. All of these things are buried deep in our cells because they're very hydrophobic. And so they get picked up by the fats, oils, and lipids in our cells, and they're very difficult to get out. And so one of the ways that we've been using is this membrane lipid replacement. So it acts as kind of a conveyor belt system where you put the lipids in and the lipids go in and the lipids come back out. And all these different lipids inside the cells are present in all the different structures of the cells. So we have various lipid structures. These could be vacuoles, mitochondria, membranes, various types of lysosomes, chylomicrons, lipid globules, lipid droplets, all these different things, these different lipids, in fact, are sinks. They're hydrophobic sinks for various toxins that enter your cells. And of course, they can damage your cell and be stored in your cells long term and very difficult to get rid of. So mitochondrial function is basically most of my talk tonight is just because I've concentrated on this subject that I put this slide up, as mitochondrial dysfunction essentially occurs in every chronic illness and most acute illnesses, and it's responsible for a number of different problems. And just to show you that you see this in vascular diseases and obesity and diabetes and all kinds of different related syndromes and so on, infections, coronary problems, obesity, diabetes, heart failure. Practically everything that you can think of involves mitochondrial dysfunction. Now, mitochondria, of course, are also important in innate immunity and inflammasomes. It provides a number of different basic properties of cells. It's important in calcium homeostasis. It's important in autophagy, mitophagy, apoptosis, you name it. It's present in all kinds of different properties that cells have, and mitochondrial function is important for those properties. So if we focus in on the inner mitochondrial membrane and the electron transport system in particular, it's made up of different complexes, and these complexes pass electrons, and in the process, they pump protons out from the matrix to the intermembranous space between the inner and outer membrane. And so the whole function of the electron transport chain is really to act as a proton pump. And when the protons, where enough of them are outside in the space, we can flow them back through this ATPase and the process generate ATP. This is how our energy is generated in the mitochondria. And of course, the mitochondria supply 90% of the energy of the cell, so it's extremely important. So if this is functioning properly, everything works fine, but often it doesn't function properly, or there's an imbalance in the membrane. It leads to the production of reactive oxygen species. And essentially, the oxygen gets converted into free radicals, and those free radicals are very reactive, and they can react, for example, and form nitrogen radicals, or they can form peroxides, or they can form hydroxyl radical, and so on. All these are very damaging to cells. And as you age, these damaging free radicals build up. And it's a natural consequence of aging, and they do additional damage. For example, they can react with many things in the cell, not only membranes, and particularly double bonds and membranes are very sensitive, but also DNA, other types of things, RNA, a variety of different structures in the cell are very sensitive to the reactive oxygen species. So as we age, these build up and cause damage, and those damage accumulates during aging. So we want to prevent that. Now, there are a number of different treatments for mitochondrial dysfunction, and they're geared to what's damaged in the mitochondria. For example, if there's loss of maintenance of this inner mitochondrial transmembrane potential, which is normally maintained at about minus 150 millivolts, and as we age, I mentioned that goes down and that becomes a problem, we can use membrane lipid replacement to replace the damaged lipids in the membrane and actually restore that potential. We can actually demonstrate that very nicely in vivo or in vitro. On the other hand, there may be alterations in the electron transport chain function, and membrane lipid replacement also helps with that because it provides a fluidizing effect on the inner membrane so that it maintains the mobility of these transmembrane components of the electron transport chain. Then they can function better, and that's absolutely necessary. It also maintains the ability of CoQ10 to transport electrons in the inner membrane, so it's very important. Also, if there's reduction of transport of critical metabolites into mitochondria, membrane lipid replacement helps with that as well. It helps with the outer mitochondrial membrane to keep it fluid so we can transport materials into the mitochondria. It helps provide the mitochondria with the proper membrane environment to actually generate ATP, and ATP is very important in transport and so on. And there are other events, of course, that are important. And I'm going to go back to chemical exposures and genetics. We know that membrane lipid replacement actually prevents the genetic damage of the mitochondrial DNA, which happens during aging, and that's also another important aspect of it. So there are a variety of treatments for mitochondrial dysfunction. Now, I've been talking about the replacement of mitochondrial lipids, membrane phospholipids with their fatty acids and so on. But obviously, there's some other things that are important, sometimes in trace amounts, but they're important vitamins, minerals, cofactors, particularly CoQ2, alpha lipoic acid for helping transport and so on. Different metabolites are important. Antioxidants are important to try and minimize this oxidative damage. And there's certain herbs that can help in this process. Now, membrane lipid replacement components, as I've mentioned, contain all the different glycerol phospholipids with unsaturated fatty acids that contain inulin, but we've also mixed in a variety of other components, and that we'll come back to in a moment. So if you just look at mitochondria, if you take cells out of a healthy person and stain the mitochondria with one of these transmembrane dyes, like JC1 or rhodamine 123, I'll show you this in a moment. But you see if the mitochondria are very healthy, the dye shows up in the periphery of the cell. All these little punctate versions of the dye are mitochondria within the cell, and they're all operating. They're fully functional. We have a transmembrane potential of a minus 150 millivolts, so everything looks good. But if you take a cell from, for example, a patient with chronic fatigue syndrome, myalgic encephalomyelitis, and you look at that, you still see it, but it's very faint. You can see the faint little deals here. So mitochondrial function is way down in those patients, and that's what really causes their fatigue. They don't really have enough energy production to maintain all their cellular systems, and fatigue is one of the most important or most obvious loss of function. So we can actually prove that there's using NT-factor lipids, that there's a direct correspondence between fatigue and loss of mitochondrial function. So this is a crossover clinical trial. We've looked at mitochondrial function in subjects and their leukocytes. So we've taken white blood cells out of their system and looked at them, and you can see by staining with these mitochondrial dyes, you you can actually see the functional mitochondria, and you can see that by an increase in the fluorescence. And as you see with when they get the anti-factor lipids, with time there's an increase in mitochondrial function, and it's in these patients it was about half as much as what we find in a normal healthy 30-year-old, and these are all older people, by the way, around 70 years old. But you can see with the membrane lipid replacement, we can actually bring that mitochondrial function up to the level of a much younger person. Just it takes time. It's dose dependent. We've gone to much higher doses and it takes less time, but in this particular experiment, we use a moderate dose. And if you look at fatigue in those patients, the fatigue goes down accordingly. So as the mitochondrial function went up, the fatigue went down. And so this is results of fatigue testing on those same patients. Now, in these patients, this was a crossover trial. So the same patients were crossed over. If they were on placebo, they were crossed over to the actual supplement, the anti-factor lipids, and vice versa. So in the crossover part of the trial, if we switched them to the placebo arm, guess what happened? As you'd expect, it slowly it decays, and the mitochondrial function slowly goes back down again. And along with that, of course, the fatigue comes back up again. So there's a direct correspondence between the mitochondrial function and fatigue and the ability of the anti-factor lipids to restore mitochondrial function. So we've done a number of trials on this, and this to show 4 of the 7 trials that we published on. We can look at some of the results in aging patients, average age 70 years of age. There was a 35% reduction in fatigue, very significant. And in patients who were moderately aged that had complaints of chronic fatigue, but they didn't fulfill a full criteria of chronic fatigue syndrome, they had a 40% reduction in fatigue. In fibromyalgia patients, which is a fatiguing illness, and chronic fatigue syndrome patients, there was a healthy reduction in fatigue. And it's just, here's another aging and fatigue trial. So in all those trials, we found a reduction in fatigue. I'll show you a little more detail in a trial on fibromyalgia patients. We've gone to a much higher level of anti-factor lipids, in this case, 5 grams per day of the anti-factor lipids. In this case, there was a little bit of controlled release caffeine that we thought that might make a difference. Turned out it didn't make much of a difference because in a subsequent trial, we left that out and there was virtually no difference in that. So it's essentially 100%, 95% of the effect is due to the anti-factor lipids. So in this short-term trial, so this is only about a week or so, there were overall symptom improvements of about 36%. There was a reduction in fatigue of about 38%. Reduction in pain, I'll come back to pain because I've been concentrating on pain more recently because there's a very interesting effect that anti-factor lipids has on the actual ion channels that control pain. And all these ion channels have lipid binding sites, and so the lipid environment around the ion channel turns out to be very important. And you have to essentially have fluid lipids there, or otherwise they don't function properly. There was a reduction in gastrointestinal symptoms, very dramatic reduction, and overall improvement of quality of life indicators. Now these are just showing the, oops, I'm sorry about that. Let's see if we go back. So the overall all symptoms were reduced very dramatically. There was a reduction in pain, reduction in fatigue, and you see it happens very quickly, a little over a week or so, reduction in fatigue, reduction in GI symptoms, and in this case, there's improvement of quality of life. In this case, the way the QOL tests were done, there's less quality of life if the bar remains high. There's more quality of life if it actually goes down. So in all the cases, we found a reduction, very significant reduction in a lot of these parameters associated with quality of life. So we found in a number of different clinical trials that the membrane lipid replacement restored mitochondrial function. We saw this in aged patients. We saw this in fatigued patients, adults that complained of chronic fatigue, but they didn't fulfill all the criteria for chronic fatigue syndrome or fibromyalgia syndrome or other fatiguing illnesses. So it corrected aging effects, and it did this by restoring mitochondrial function. And also, we were talking very early, it was endothelial cells were brought up. Well, this membrane lipid replacement is very advantageous to endothelial cells, and it's very important for fluidizing the endothelial membrane, which is very important in the coronary conditions and maintaining proper vascular flexibility and so on and so forth. So we can come back to that later. We've also found in studies which I didn't really present that along with reducing fatigue, there were improvements in short-term memory, mood, executive functions, and other cognitive abilities in patients. And these are mostly done with older patients where it's pretty obvious what's going on there. And they've benefited quite dramatically in this. I mentioned that the membrane lipid replacement provides important precursor molecules for a variety of different things. You were talking about hormones. Some hormones require these membrane glycophospholipids as part of the precursor molecules that help restore them. And ion channel functions, I'll come back to that, absolutely essential in modulating pain and modulating some of the peripheral effects that happen and go haywire during various diseases and with aging. We'll come back to that. I'm working now with things like peripheral neuropathies and other types of symptoms, peripheral symptoms. Also, various clinical symptoms are reduced. And I'll show you a clinical trial where we've looked at over 100 types of symptoms, so we know what's benefited pretty precisely with membrane lipid replacement. And finally, I'll end up at the end of my talk with the removal of hydrophobic toxic molecules from cellular stores. We were talking about plastic precursors, for example. All of these things are quite hydrophobic, and the way you can remove them is with the membrane lipid replacement. It's a very slow process. So now I'm going to talk a little bit about these multiple chronic conditions. One is Gulf War illness. I do a lot of work with veterans, and I've worked with primarily two types of problems because of their exposures. One type of problem were chemical exposures. They were exposed to a variety of different chemicals. I've worked, for example, with petrochemical exposures and presumed chemical biological warfare agents, but there were other types of exposures as well. For example, to really insecticides and other things that they were sprayed on their clothing and so on. A few of them had radiologic exposures. I won't talk about that, but they were very few, mostly depleted uranium that were in the munitions that were used during the Gulf War. And some of these people were exposed to some of the munitions or the dust that these munitions cause when they penetrate an armored vehicle. That dust is very toxic, and you can inhale it and get U-235 in your lungs. It spreads to your bones and so on. That's another problem that we're not going to talk about tonight. Biologic exposures, there were some various types of infectious diseases that went on in the Gulf. And importantly, one of the problems that I worked on almost exclusively was the fact that they had mycoplasma, and about half of these veterans came down sick. And that was due to the fact that the mycoplasma was actually a contaminant in the vaccines that they received during their deployment. And this is why so many of them came down sick, even the ones that were vaccinated and were not deployed, or were vaccinated and deployed were 100 miles, 200 miles away from the actual front lines and so on. So they weren't anywhere near the battlefronts. And of course, there were environmental exposures and sand, dust, and fumes and so on. So this complement of chemical exposures and environmental exposures and biologic exposures form these sort of multi-factorial diseases called Gulf War illnesses. And so basically they're a family of different chronic medical conditions. And so we've studied both these. I mentioned that I was involved in, particularly my late wife and I, in determining that mycoplasma was a major problem in many of these veterans, and we devised some treatments for them, and those infections spread to family members. And in the cases where the families became involved, almost 100% of those were due to the mycoplasma spreading from the Gulf War veteran to the immediate family members. So we worked on that problem. Tonight, I'm going to talk about the chemical exposures were caused by pollutants, and most of them were actually caused by very heavy exposures to fuel, exposures to burned petrochemicals. There were oil well fires. There were about over 100 oil wells that were blown up by the Iraqis when they retreated. These caused tremendous amounts of oil and fumes and burned oil well products to be released in the atmosphere. And they breathed these in, and these were causing major problems. And so we're going to talk a little bit about those types of exposures. Now, I mentioned pain because the pain is something that membrane lipid replacement we found is good for, requires a higher doses of the membrane lipid replacements. We know the reason for that now. It's because the actual ingredients in the membrane lipid replacement that are important in inhibiting pain by binding to certain ion channels in the nerve membranes are basically present in very small amounts in these membrane lipid replacement lipids. And that's why we needed the higher doses. We'll come back to that. But we had a number of case reports on membrane lipid replacement helping patients. And one of those I'm going to go into, I'm not going to mention it, but just to say that this is what really piqued our interest, because we looked at some gunshot cases from the Gulf War, wounds that never healed from the Gulf War. And by giving a membrane lipid replacement, we finally got those wounds to heal. And so these were wounds that had been stayed a major problem in these veterans for decades. And we finally managed to make some progress on that. So there's a case report on that. And also we've started this by looking at the fibromyalgia symptoms in the Gulf War veterans. We found that after 4 weeks of membrane lipid replacement with higher doses of anti-factor lipids than we'd use in our civilian cases, we found that there was a dramatic reduction in fatigue using different types of ways to measure fatigue, the Yale method or the Wisconsin method of measuring pain. Doesn't matter, we got almost similar results with the same types of studies. But the reduction of pain in fibromyalgia patients and in Gulf War illness patients by membrane lipid replacement was really interesting and could be caused by a number of different events. It could have been caused by normalization of peripheral nerve resting potentials. We do know that one of the reasons why pain is transmitted and keeps being transmitted over and over, so chronic pain, is because the resting potential of the peripheral nerves is interfered with. It comes down. It's not maintained properly, and therefore those cells are much easier to fire. So those nerve cells are firing all the time, and it causes a chronic condition called chronic pain. And there are also effects on the nerve transmission as well, not only just the depolarization, but the actual transmission of pain depends upon the transmission across the synapse. And membrane lipid replacement is actually important there as well. I'm not going to get into that tonight, but there are also other effects on nerves. Peripheral inflammatory cytokines. Membrane lipid replacement actually reduces those peripheral inflammatory cytokines. That has a very positive effect. I mentioned the effects on peripheral circulation, and the microcirculation is very important in terms of chronic pain, and it's very important to have fluid membranes in the endothelial cells in the microcirculation. Otherwise, if they become too stiff, this presents a situation where the microcirculation can cause problems with the actual nerves and so on. And then we found that the membrane lipid replacement repairs gastrointestinal permeability, and the so-called leaky gut problem is a major problem with people that have chronic pain symptoms. And the membrane lipid replacement helps repair gastrointestinal permeability by repairing the epithelial lining of the gut and repairing the endothelial cells that line that. All of that's very important. Now, we can show that in culture very nicely. If we take dorsal root ganglion and put them in culture, they're shown here. And this was done in collaboration with the Ion Channels Laboratory in the Department of Biophysics. my collaborator, Professor Gonzalo Ferreira in the University of the Republic in Montevideo, Uruguay, and he and I have been longtime collaborators on various things, and he's kind of an expert on nerve transmission and studying this in vitro. And I just presented this recently. And so just to show you how this works, we can take nerves in culture and we can present them with a ramp depolarization. And if we ramp depolarize that field around them, we can get the depolarization of the nerves. Here's the depolarization of the nerves, and then as they readjust and come back to control levels and so on. If we put in membrane lipid replacement, in this case as nanomicelles of these lipids, these membrane phospholipids, we can attenuate that. So instead of the ramp depolarization, now it's modulated. We don't get the same depolarization. And we can actually, by adjusting the types of lipids, the amounts of lipids and so on, we can adjust this so that we can actually control the depolarization process and the firing of nerves and cultures. This is a neat way of showing how membrane lipid replacement can control nerves. And it's probably, to the most part, due to these transmembrane nerve channels, in these cases, the TERP channel, the TERP family of channels, which are very important in nerve transmission. And these are the channels which control the flow of ions in and out of the cell. And if they have the correct lipids in binding to those channels, they can function. But if you replace those lipids with our membrane lipid replacement, we can affect those channels and basically bring them back under control. And this is shown here in a study we did of Gulf War veterans. Practically all of these veterans were chosen because they had severe problems with pain. They had other major symptoms that they reported. And these were after chemical exposure in the Gulf. And most of these patients were exposed to fuel oils and fuel from oil spills and oil well fires and exposure to burn pits. See, essentially 100% of them. Some of them were exposed to pesticides or components to counter, excuse me, I went too far on that, to counter chemical warfare. Let me see if I can go back here. And some were exposed to insects or sewage or so on. Some were actually exposed to chemical and biological weapons, but those were very few compared to the exposures to oil well fires, fuel oil, burn pits, and so on. So most of these patients had those exposures. Now, we look at symptoms categories. I'm going to show you some symptoms categories. And basically, these symptom categories are made up of symptoms. And we look at about 100 or more signs and symptoms when we assess these patients. And these are grouped together. For example, we might group breathing, for example, in unable to breathe deeply, wheezing, shortness of breath, other breathing problems, or neurologic, depression, loss of interest, suicidal thoughts, mood swings, memory loss, headaches, concentration, and so on and so forth. So these are based on a lot of different criteria. So here's some of the results we got with our veterans when they went on membrane lipid replacement. So we gave them a fairly reasonable dose of this. They got actually 6 grams per day of membrane lipid replacements. And we looked at a number of functions that we based together. So these are heart functions. And we're just going to look here basically at the red lines. These are 2 individual patients to show you the variability. So the blue here, the purple and the green are 2 individual patients and their responses. The red here are the averages of all patients and the significance of that. So, for example, heart, nasopharyngeal symptoms, breathing symptoms, neurologic symptoms, sleep, and these are basically sleep problems going to sleep, problems staying asleep, other sleep difficulties, fatigue problems, and I've done a lot of work on fatigue, and I showed you some of the results on fatigue. You see there's a nice reduction in fatigue with membrane lipid replacement. infections, gastrointestinal problems, urinary problems. And on the next slide, go to the next slide, visual problems, balance problems, sensory problems, audio problems, muscular problems, and so on. Chemical sensitivities, very important. And we found that that was in pain. And the pain was very interesting because I'm interested in that from the normal of different directions. So membrane lipid replacement is a functional approach to repairing cellular membranes and in the process of reducing symptoms and restoring fatigue. And we've looked at it in terms of general health. We've looked at it in aged patients. As they age, of course, a number of things tend to start to go wrong, and you can correct some of this with membrane lipid replacement. So we've looked at fatigue and a number of different conditions. We've looked at inflammation. We've used this actually in weight loss and obesity because of the very important effects that membrane lipid replacement has on actually attenuating the need for high fatty and high lipid foods. So it actually cuts the craving for that in a very interesting way. We've looked at the things associated with menopausal fatigue, brain health. I'm not going to go into that. Cardiovascular health, very important. Metabolic syndrome and diabetes health, very important. And fertility, fatigue. I told a little bit about fatigue. And we've used this during various types of injury now. And we've most recently used this to help repair gunshot wounds in veterans. If wounds fail to heal for virtually years, and we've been able to heal those. The veterans finally their wounds are finally able to heal. And finally, down at the bottom here, chemical detoxification in Gulf War illnesses, to use it to detoxify chemicals, because the membrane lipid replacement will slowly partition chemicals in from the stores that they get concentrated in cells. And this happens when we age as well. And it will slowly remove them by continuing to partition them to other compartments in the cell, and eventually to the lipids which are outside the cell, and eventually remove them from the cellular environment. So membrane lipid replacement, we found, increases mitochondrial function. We found this in aged subjects and also in fatiguing illness patients like chronic fatigue syndrome, fibromyalgia syndrome, Gulf War illnesses, and so on. All these patients benefited in terms of their fatigue by taking the membrane lipids. So it decreased fatigue, pain, and other symptoms in aging subjects and cancer patients, for example, chronic infections and chemical exposures. It reduced homocysteine levels, which are, homocysteine again is a marker in these systems, and it's a marker for future heart attack and loss of life. And we were able to reduce homocysteine levels. And very interesting study we did on homocysteine. Those patients are still doing well, and they had homocysteines in the danger level, which would predict a heart attack. And yet we put them on the membrane lipid replacement. We've had them now, some of these patients, for years on membrane lipid replacement. They haven't had a heart attack yet. So that's a really interesting finding. We were able to show, and I didn't show that, we were able to show that we could decrease deletions in mitochondrial DNA in aged subjects. And this is a hallmark of aging. We could decrease the effects of cancer cytotoxic therapy, very important. And I came from the MD Anderson Cancer Center before I retired. I was the department chairman, and one of the things that we were very interested in was trying to reduce the cytotoxic effects of cancer therapy. And this is one of the things that does that very nicely. And it reduces other symptoms, cognitive symptoms, gastrointestinal symptoms. It helps with sleep and other things. It slowly reduced symptoms in chemically exposed veterans, and it's very well tolerated. So that may have been, that was not my last slide, but it was close enough. And I think I'll just end the talk there, and just to say that one of the things that we've been very concerned about through all our studies is to make sure that membrane lipid replacement is absolutely safe. And I can report to you that there are virtually millions of people have taken membrane lipid replacement supplements without any indication of toxicity. So it's completely toxic free. In fact, it reduces some of the symptoms, the side effects of chemotherapy we found most recently from other agents. So not only is it beneficial, but it actually helps in other types of therapies as well. So that's basically one of my presentations, one of several presentations that I have on using membrane lipid replacement for various types of problems. But this is kind of a nutshell, gives you an overview of some of the things that we've done with membrane lipid replacement. And I'd be willing to answer any questions if you have any.
I'd like to ask one. Is the glycerol phospholipids you're referring to, is how similar or different is that from plasmalogens?
Very similar to plasmalogens. The difference is really in the makeup, in the composition. The membrane lipid replacement is basically a souped up version of plasmalogens, in that there are certain types of lipids that we find are higher in abundance. For example, like phosphatidylserine, for example, are certain things that are important that we find in membrane lipid replacement that makes it more suitable for synthesizing really important molecules in this nervous system. For example, things like cardiolipin are extremely important in the vascular system and in the nervous system. And membrane lipid replacement provides the precursors for that in elevated amounts. And it really helps tremendously in some of the symptoms.
Dr. Nicholson.
Yes.
Hi, Dr. Ron Klotz here. A great presentation, very interesting. Did you look at nerve conduction velocities in people
We're
with?
We're just beginning to do that now, actually, and we've taken a look at velocities in ex vivo hearts, for example, the model we've used. So we put ex vivo hearts, you put them in a heart bath, and you connect them, and you innervate them, and then you can look at a variety of different things. We can look at innervation. We can look at heart function. We can look at a variety of things, and that's one place that we're just beginning to do that. And so
it's very easy. It's very easy to test neuropathy for testing like the sciatic nerve and other long nerves for the velocity. It would be very interesting how this works for that. Also, an easy test, which would be interesting, would be a P300. Because are you seeing improvements in cognitive function?
Yes, we have, and that's one of the interesting things you see with the membrane lipid replacement is that people do better on various cognitive tests and so on. It takes a while with membrane lipid replacement, but I think two things are happening there. Number one, we're supplying some of the lipids that brains really need, and the other thing is we're removing toxic molecules from the brain, but it's a very slow process. So it can take up to a year to remove toxic molecules and so on.
Can we talk about
how expensive is this?
Can we talk about the money and all that stuff after we close the CME, please? The cost.
Hi, this is Sylvan Garth. Hi, I have a question about the membranes are mostly unsaturated glycolipids is what you're adding, correct?
No, they're a mixture. They're a mixture, actually. But it's the unsaturated lipids which often form the annulus around proteins, and that's why they're so important in terms of allowing protein function. And so that's very important. And in the membrane lipid replacement, we have a lot of unsaturated glycerophospholipids that go into that. So that's an important part.
I'm wondering if you load those membranes lipid into cyclodextrin, that's a delivery and stabilizing. So the cyclodextrin will deliver the healthy lipid membrane and then collect the bad ones, oxidize it, damage it, lipid membranes.
Well, here's what we found is that when we go to higher doses of membrane lipid replacement, so we're talking in 6 grams per day or more, this happens naturally because the lipids themselves will naturally pick up the toxic molecules and transport them back out in the reverse process of the way they're transported in. So this conveyor belt model that I put up is operating in both directions. So it's both bringing the right lipids in and taking bad lipids out. It takes a while. It's a very slow process of partitioning and removal because of all the different lipid compartments that are involved, but eventually we can slowly remove them, and you can actually, in the case of the Gulf War veterans, for example, they were actually smelling some of the contaminants in their stool that are coming out. They could actually smell them, particularly some of the more dangerous ones. So they knew it was working when they could smell the chemicals in their stool. So
Well, we have proven that cyclodextrin does clean the bad lipids from the blood vessels. And we have a lot of patients, including some of them here, they will witness that. But if,
yeah, I'm not saying
it's,
that this is the only approach.
yeah,
What I'm saying is that this causes a variety of different actions besides just simply detoxification. It replaces the important molecules and provides precursors, for example, for cardiolipin in the mitochondria, absolutely essential. Cardiolipin is for the health and ability of the mitochondria to be fully active. And this provides the precursors for that, and as well as removing some of the toxic materials from mitochondria and other stores. Eventually, the stores inside cells and are full of contaminating chemicals and so on. And we can bring those out, but it's a very slow process. You don't want to mobilize them too quickly because patients can react to that if you mobilize their toxins too quickly.
Dr. Nicolson, have you experimented at all with COVID vaccine injury or long COVID people that
yes.
had?
Yes, we have. We use this.
From that?
This is an important part of a long COVID treatment, as a matter of fact, to give these membrane lipid replacement molecules. And so, yes, absolutely.
All right. My next question is, in trying to look for severe lipid membrane damage, have you looked into testing autonomic nervous system for indications of that?
Well, we're just getting into nerves, as I mentioned. And we're getting into this slowly because the first thing we're doing is we're looking at mechanism as well as trying to look at various nerves to see what the effects cause and can we explain those effects, because it could be from a number of different things.
And the deal function test will be the best test to measure, because if your membrane replacement therapy helps to increase the flexibility and liability of the blood vessels, then you will see the vascular, you can see it instantly. I mean, improvement.
I didn't want to talk. We have some preliminary evidence on that, and the answer is yes. It improves the flexibility of the microvasculature. I want to get into that because I haven't published a paper on that yet. Everything that I talked about today is published.
This is Sylvan again. I have an important question. On the same program, we've had a lecturer saying, as you age, you want more saturated fats in your lipid membranes because it makes them stronger, safer, and less prone to damage, which is exactly the opposite of what, I mean, you are the guru on membranes for years.
Well, if you talk to
there's a C15 company.
If you talk to nutritionists, you talk to nutritionists and you get the exact opposite response to what you just said. They would say that fluidizing lipids are much healthier than non-fluidizing lipids, particularly as you age. So that's a controversy that's been floating around for a number of years and so on and so forth. I haven't worked as much on aging, but I can tell you that we're very interested in this, and we're doing some work on this, and some of the aspects of aging that, for example, the buildup of toxic materials inside cells and so on. This helps with that. It's a slow removal process, which is beneficial because it doesn't trigger the reactions, the severe reactions you get sometimes when you remove toxic substances too quickly. So that's a benefit, it turns out. And we're just getting, involved in this. So,
but it was just confusing to me because I've been following you for 40 years now. But there's some strong people out there, including on this program, were saying no, you got to replace them with saturated
Let me go ahead and comment on that, okay?
Yes,
the
please, because
problem-
I'm confused.
The thing is that I think we understand that the fat itself is not the problem. We need fat, we need cholesterol in our body. Whereas the problem is oxidation of those fat.
Yeah.
It's
and I
the
agree
bad
with that.
thing. And
I
that
agree with
oxidation
that.
comes from sugar, because sugar gets oxidized and become glycans, and they become free radical generators, and they will oxidize those fat, whether it's fats saturated or unsaturated, and they become oxidized LDL, and that's the damage. So as long as you control the sugar, and of course you don't want to have too high fat in your blood too. You know, people with family hypercholesterolemia, we need to find a way to decrease the level of the cholesterol back to normal, but not low. If you go low, like what they do with statin, too much, to the point they don't have enough cholesterol to make sex hormones, then you end up again with oxidative stress and damage of the blood vessels. And we are now creating something that helps to clear up extra fat and oxidized fat with cyclodextrin. We're going to talk about it after we finish the CME. But again, I think cyclodextrin loaded with your lipids, I think it's the best option. So you can do both. You can clear with the cyclodextrin all the bad fat, dump into the bile, at the same time replenish the lipids membrane, cellular lipid membranes with healthy lipids. Something you may consider, and I will send you some publication to support that. But let me add something here very critical. We have been 1 month away from CME because of the technical issue. Now we got the CME back. So I want everybody here, if you are interested, go ahead and get your CME from the link here, which I'm posting it here. Do we need to close the CME so we can be more freely talking? And
Boy, this is Sylvan again. You're
so
basically bypassing what
close
I
the
asked
recording.
you.
Sylvan,
Here-
let's
here-
close the recording. Can we close the
Okay.
recording now?
Yes, because, okay.
Recording, is that